Hi, good afternoon to the Asthena talk by Lukas Landegger. He is medical doctor and scientist at the Medical University of Vienna, currently working in translational research, and he's a resident and otolaryngologist at the Medical University of Vienna. He graduated from Innsbruck, another medical university in Austria.
That is likely to make the foundation very happy.
Outstanding research. I apologize for the interruption, but we are getting started now. He served as a military doctor, and his research now focuses on hearing loss. He did research and served as a military doctor and as a scientist in several countries like the United States, the UK, France, Spain, Germany, Singapore, and Australia. And he's an award-winning and well-funded, outstanding scientist now, focusing on current sequencing technologies that allow us to provide a fast compendium of growing genetic alterations that we can see and understand now that underlie human hearing loss. He will explain what kind of targeted therapeutics we can apply, such as gene therapy and other pharmacological therapies that are upcoming in order to treat hereditary hearing loss, but noise-induced hearing loss and other malignancies. Lukas, the stage is yours, and please feel free to share some slides with us. It's a very, very exciting topic and an outstanding topic in translational research.
Well, yeah, thanks a lot for having me. I'll try to pull up the slides.
Great.
I think you should see them now. Exactly, perfect. So yeah, title of my talk is inner-ear gene therapy. The majority of the research that I'll present today was carried out in Boston, but we are doing a lot of follow-up work here in Vienna now. Just as a start, we do have potential conflicts of interest or theoretical conflicts of interest. A lot of these studies are funded or in collaboration actually with industry because there's a big interest here, and that was already in Boston and now still in Vienna. We actually managed to obtain a relatively large fund, so that allows us to establish a Christian Doppler lab here in Vienna now that started in July together with my former mentor, Christopher Arnold now. And just to give you a presentation outline now, I'll start with an overview regarding hearing and the inner ear in general. Then we'll talk about inner ear gene therapy or general gene therapy. Afterwards, we'll focus on adeno-associated viruses in the mouse, after which we are going to AAVs in the primates, so in the monkeys. Then I will introduce you to the optical cochlear implant, which would be a very neat device in the future that hopefully helps patients to hear even better. And subsequently, we are going to speak about potential patient acceptance. So what do patients actually think about all these novel therapies that are coming out? And then we will briefly discuss the remaining clinical translational barriers. So what is actually needed to bring all of that to the clinic. So why should you care about hearing loss? Hearing loss is actually the most common sensory deficit in humans, and the WHO released a statement in April 2021 where they summarized that over 5% of the world's population has disabling hearing loss. And that is actually expected to rise even more to eventually reach over 700 million people by 2050. And in addition, over 1 billion young people are actually at risk of hearing loss due to unsafe listening practices. So think iPod generation. Nowadays, everybody runs around with music in their ears all the time. And the reasons for hearing loss do not only include noise exposure, but all of these different reasons as well. And if we look at the age distribution, then we can see that the older we get, the more likely it is that we develop hearing loss. And eventually at the age of, in the age group between 85 and 90, we actually have a percentage or a 72% chance of actually suffering from hearing loss. So how does hearing work? If we look at this slide, then we see that the external ear is out here. So sound comes through the external auditory canal, then hits the eardrum, and then we have the 3 ossicles in the middle ear until we eventually reach the fluid-filled inner ear. The ossicles, the last ossicle is actually the tiniest bone in the human body, the stapes. And then if we zoom in here, then we have the cochlea, the snail-shaped organ here that actually allows hearing, and the vestibular system here. So we have the saccule and utricle that are responsible for horizontal and vertical acceleration respectively, and the semicircular canals that are responsible for rotatory movements. And so once the sound comes in here with a certain frequency and with a certain loudness, then a wave is created. And depending on the frequency or specifically the frequency, certain cells are stimulated along the length of the cochlea. So very high pitched sounds are stimulated down here at the base, and very low pitched sounds are stimulated up here at the apex, so at the top of the cochlea. And the cells that are in there, the sensory cells, are the so-called hair cells. And once those are deflected through that traveling wave, they actually stimulate the auditory nerve, the vestibular cochlear nerve here, the 8th cranial nerve, and that then forwards the impulse to the brain where we kind of then hear. So what can we do at the moment if patients have hearing loss? So if it's not that, we're talking about sensorineural hearing loss, so specifically hearing loss of the inner ear or structures behind it, not so much the middle ear where we have a lot of different other types of prostheses that we can put in, and it's relatively easy to fix overall. It's a simplified statement, but however, sensorineural hearing loss, we don't really have anything except for hearing aids if the degree of hearing loss is not that severe. And if we're close to deafness, then we're talking about a cochlear implant. So what is the second part, this cochlear implant? We basically have an external sound processor out here that detects the different frequencies and loudness levels. And then we have an internal implant here with an electrode that goes all the way into the cochlea, so into the inner ear, into the snail-shaped organ that we just discussed. And what this does, it basically that it replaces the hair cells, because depending on which stimuli are being detected, different areas here at the level of the cochlea are being stimulated with electricity. So this electrode then sends out electricity to stimulate the cells that eventually form the auditory nerve. And it's really remarkable that this works. It's the most successful neuroprosthesis in general. And however, there are a few shortcomings that we'll discuss later. And then I'll give you a brief introduction regarding gene therapy. So what is gene therapy? If you look it up, it says the transplantation of normal genes into cells in place of missing or defective ones to basically make those cells function again. And specifically related to hearing, there's over 120 genes that cause non-syndromic hearing loss. If we compare otolaryngology with ophthalmology, so with the eye doctors, then we have to say that we're kind of lagging behind, because there's actually dozens of active gene therapy trials for various diseases of the retina. However, there's only a handful of active trials for severe to profound hearing loss. And if we talk about this AAV specifically, so these adeno-associated viruses, then there's actually already licensed therapies for a specific type of blindness, and Luxturna is the brand name for that one. And you might also have heard of Zolgensma, which is actually at the moment the most expensive medication in the world to treat this horrible disease of spinal muscular atrophy with remarkable results. And this is just an overview of how viral mediated gene therapy works. This is an adenovirus. My research specifically focuses on, or primarily focuses on, adeno-associated viruses, but the mechanism itself is relatively similar. If you're interested, we can talk about the differences later. But basically, you bring the vector to the cell that you want to transduce. So that means to infect, basically. And then the vector is being incorporated into the cell and then eventually delivers its cargo into the nucleus. And that's then where the cellular machinery kind of produces the protein that's required. And through that, the cell should hopefully be working again. So what are simplified strategies and how would that work if we think of genetic diseases? So basically, if we have a recessive disease, which means that every gene that we get, we have basically one copy from your the parent, from your father, and one copy from the mother. And in recessive cases, we have no functioning gene, so we inherited two genes that do not function from both parents. And then in that case, actually a simple gene addition would be sufficient. So if we introduce the functioning gene with a virus, then that should hopefully work to actually get the cell running. And that is what most animal studies so far have done. And we'll talk about a few of those in just a second. However, we can also hopefully target dominant diseases by a so-called gene disruption, because then we have one gene that, one allele specifically, so one inherited part of the, or inherited gene from the one parent. And then what you can do here is that you try to just disrupt that gene, so destroy the gene, and then the other gene is taking over. So the other gene copy is taking over. And then because that is normal, then hopefully the cell should work again. And then we could carry out specific gene editing, where you probably have heard of CRISPR-Cas9, which for the discovery of that, the Nobel Prize was awarded. And what you can do with that is to specifically cut out certain sequences and then try to replace those. There's another type, but we won't really talk about that during this talk. So if we think about gene therapy in the human inner ear, or inner ear in general, then we'd have to get the vector in there somehow, because the inner ear is secluded kind of from the rest of the body. There's similar to the blood-brain barrier, there's a blood labyrinth barrier. And the idea is that the idea that most people have followed so far is to actually carry out an injection through the so-called round window. And that is relatively promising in terms of translational approaches, because this is also the approach that cochlear implant surgeons these days have to, or use most of the time, to introduce the cochlear implant into the inner ear. And so that's, a lot of people could carry out this surgery if those therapies were to become available. And we have 3 rows of outer hair cells that have an amplifying function, and 1 row of inner hair cells, and that's that specific type of cells that causes the switch from the mechanical to the electrical impulse that I mentioned before. Once those cilia at the top of the cell are deflected, the switch or the stimulation of the auditory nerve then occurs. And what we're really kind of striving for, ideally, is to restore natural hearing, because if those cells don't degenerate in the first place, then hopefully the kind of hearing that the patient eventually benefit from would be really close to the normal hearing. And one of the first studies that looked at this was this here. This group used VTLA3, so a specific glutamate transporter that's important for the communication between these inner hair cells and the auditory nerve. And they created a knockout model, and those mice are actually deaf. It's not a very common issue in humans, so it's only relevant for a few human patients, but it does exist. And what they did was that they used an AAV1 to target those inner hair cells, and that's exactly what is needed here. And what you can see here with these curves, so that's actually a so-called brainstem evoked response audiometry. So you present a stimulus at the ear, and then you measure the electricity at the level of the brainstem. So kind of similar to an EEG, although not exactly the same, but those are the waves that you get. And then if you have these curves at the level of the brainstem, then you basically, sorry about that, then you basically can say that the stimulus was processed and there is a response. In the knockout model, you see that there's no response at all. So those mice are completely deaf. However, once these knockout mice were injected with this virus carrying the corrective gene, you see that these mice heard again. And that's really remarkable. Here it is quantified, and those animals pretty much were at the same level, or some of the best animals were at the same level as the normal hearing mice. And if we look at these earlier studies, then what is kind of obvious is that all of these focus on inner hair cells. And why is that? Well, because if we look at these images created in mouse models, then again, we see one row of inner hair cells, 3 rows of outer hair cells. And in green, you see the cells that have been transfected, so infected by the virus. And you can program this virus in a way that once it goes into a cell, it produces the screen protein, and then you can detect it very easily. And so if we look at that, then again, inner hair cells were primarily targeted, but the big problem or the big difficulty here is really to reach the outer hair cells. So a virologist at Massainier, or Scapen's Eye Research Institute, so Harvard Medical School in Boston, Luke Vandenberg, he actually developed a virus that's called ANC80. And it's a computer model synthetic vector, and it's the predicted ancestor of the currently occurring AAV serotypes 1, 2, 8, and 9. These viruses actually circulate within the community, and quite a few people, actually about 80% of adults, have been exposed to one of these. And so the hypothesis is that ANC80 circumvents the pre-existing immunity because the natural kind of defense system or immune system has not been exposed to it and doesn't automatically neutralize it. And when they used it down here in liver, muscle, and retina, every column here is a different viral serotype, so AAV2, AAV8, and ANC80, then you see that there's a lot more green over here compared to these two columns, which means that ANC80 really seemed to perform better and transduce more cells in these organs than the standard AAVs that had been around before. But nobody had tried that in the ear. So what we did was that we used these Cochlear Explants. So for that, you actually extract the inner ear of mice, neonatal mice usually, so immediately after birth. And then you can dissect them and then get a half turn like this, where you again have 3 rows of outer hair cells, 1 row of inner hair cells, so in green. And in red, you have all the neuronal structures with the spiral ganglion neurons, the cells that eventually form the auditory nerve here in the center. And if we zoom in here, then we get a specific image, and I'll present some of these images to you in the next few slides. Just to give you an idea of how small these are, this here is one of those Cochlear Explants. So they're really tiny, and then under the microscope, you just blow them up and kind of zoom in specifically. So in this paper that was published in Nature Biotechnology in 2017, we did an in vitro screening as a first step, and every column here again represents represents a different serotype. Here, the established serotypes, and here on the right side, we again have ANK80, this novel synthetic virus. And what you can see here is that ANK80 again outperforms all of the other viruses, and specifically also transduces a lot of cells at the outer hair cell level. And if we then, so after these very promising in vitro results, when I first saw them, I actually couldn't believe them. We went into mice that were alive and injected those into the inner ear. And also in vivo, we could confirm that ANK80 seems to outperform pretty much all of the other vectors, specifically also at the level of the outer hair cells. So if we look here, then we see that pretty much all the outer hair cells are transduced, while over here, hardly any hair cells are being transduced. We did a bunch of other experiments with these animals, patch clamping, etc. A lot of people were involved, and we saw that once the virus was in the cell still could work the way it should, basically. Those were all healthy animals, so no knockout models there. And then we just got a lot of beautiful images in these mice, and you can appreciate even more here that we got close to 100% of inner and specifically also outer hair cells, which was kind of the big breakthrough. Even contralateral side, so even the other side, did get a lot of transduction, which was also not seen with any of the viruses that had been used to date or until that point. And also what we did was that we looked at the vestibular epithelia, because there's a lot of vestibular diseases where there's also no treatment at the moment. And specifically some of these degenerative diseases, some of these genetic diseases also include vestibular degeneration, not just hearing degeneration. And what we saw here was that also, the vestibular level, we had a lot of cells that were being transduced. And what was really great from a translational standpoint was that even human tissue that you can obtain during some surgeries, when you have to actually drill through the vestibular system of patients to remove some tumors, for example, in the inner auditory canal. Even in those, we had excellent expression, so it really seemed to be a promising candidate for clinical studies. And then a collaborating group in Boston, Jeff Holt's group, actually used this ANK80 in a mouse model of Usher syndrome. Usher syndrome is a horrible disease. It's actually the leading cause of deaf blindness, and it is one of these recessive diseases that we had just talked about. So here we have electron microscopy images, and on the left side, you can see the electron microscopy results of a healthy mouse. So you see 3 rows of outer hair cells, 1 row of inner hair cells, totally organized along the length of the cochlea. Here in the center, you have Usher syndrome. So you see degeneration of many of these cells, disorganization everywhere. And here on the right side, you see this knockout model that was injected with the corrective gene encapsulated in ANK80. And you see that most of these cells, or many of these cells, are actually rescued. And some of these rescued mice then actually heard the same as the normal mice, which was really remarkable. And ever since, there have been quite a few other studies with therapies of a lot of different recessive diseases all around the world. And these are just some of the investigators that carried out these studies. But what about dominant diseases that we had already talked about? So another group at Mass Eye Near used a so-called Beethoven mouse model. It has nothing to do with Beethoven, because Beethoven actually had something completely different in terms of his hearing loss. But what they did was that they used a Cas9 guide RNA complex that was then delivered in vivo. And what you can see here is that these are the uninjected animals with this type of deafness. And you see that there's a degeneration specifically at these frequencies down here, while with the injection, you could actually save a lot of that. That is just as a comparison, the healthy animals, how the hair cells would have looked there. But yeah, that was really remarkable. For the first time, they showed that you could also treat a dominant disease, and that was then published in Nature. And here at the level of the hearing results, they also showed an improvement. However, it wasn't as, so the louder it gets, or the higher it is, the louder it gets. And you saw that, or you see that they're not comparable to the normal animals. However, there is a significant increase in terms of hearing, or improvement in terms of hearing. And then another group at Mass Eye Near, so Harvard Medical School, Charlie Lieberman's group, actually carried out these injections in adult animals, because so far we had only done that in neonatal animals, and neonatal mice actually are born deaf and humans are not. So we were specifically interested in what happens at the adult, in the adult animals. And those were 7-week-old mice, so kind of young adult mice. And you can see that, again, we did get a lot of outer hair cells, however, not throughout the whole cochlea. So that was a difference compared to the neonatal animals. And what we also saw in these neonatal animals was actually that once we injected the virus into the inner ear, we also saw some transduction at the level of the brain. So specifically at the cerebellum, these Purkinje cells here did take up the green fluorescent protein as well. And that is something that is something that has to be clarified before, or had to be clarified before going into human trials, because obviously if there is some delivery of the virus into the brain, then that would have potentially more severe side effects compared to a virus that just stays at the level of the inner ear. And in rodents, it is known that a structure called the cochlear aqueduct is relatively patent. So there is a flow of liquid through that. However, it is not really, or it wasn't really clear in humans, including non-human primates. So what we had to do was to actually carry out experiments in non-human primates prior to going into human trials. And this was the experimental design for this non-human primate trial. So we had 5 rhesus monkeys. 2 were injected with AAV1 with tissue extraction after 7 days. 2 were injected with ANC80, also tissue extraction after 7 days, and another one was injected after, also with ANC80, and the tissue was extracted after 2 weeks. It was the left ear that was injected. It was again a round window membrane approach. 30 microliters were injected. In the mice, we usually have less than 1 microliter actually that's being delivered. And there was another procedure called the so-called Stapes footplate vent, and I'll show you in just a second what that is. So if you look at the surgery in these animals, then it's actually very similar to humans. So you drill down to the you drill the so-called facial recess, and then you can look into the middle ear, and there you see the round window over here and the oval window with the stapes over here, and then there's this fenestration, so you just go with the pick through it. The idea is that if you inject something here at the level of the round window, then by creating this hole over here, some of the fluid would come out through here, and with that you should have a better delivery of the vector throughout the cochlea. And this was this custom-made device with which we injected into the round window. So what did we learn from these experiments with the adolescent rhesus, or in the adolescent rhesus macaques? There were fortunately no obvious signs of adverse effects, no toxicity that could directly be seen. One animal actually had a unilateral left-sided facial paresis, which can occur during the surgery, also in humans, during any type of ear surgery, basically. So we did attribute that to the surgery and not so much to the virus itself, or not at all to the virus itself, actually. Also in the blood work that was done, we did not see any toxicities. We also were looking for so-called neutralizing antibodies, because if you are exposed to these AAVs, then antibodies, as we briefly had talked about before are being generated, and we saw those in serum, so in the blood, but not in the CSF, which again is promising because that likely means that the virus is not going anywhere near the brain, and really just stays at the level of the inner ear. Also in the histopathology, we did not see any apparent toxicities, and then we decalcified the inner ear and again carried out this immunohistochemistry that we already saw in the mice earlier. And in one of the 5 animals, we did not see any GFP. We again think that this was probably a surgical issue, not so much that the virus didn't really work. And here we did not see any GFP in the contralateral cochlea. All of this was published earlier this year in Nature Communications. And what you can see here is that we really extracted a lot of tissues to make sure that we're not overlooking anything. And again, we did not see in any of this histopathology any toxicities, which was very promising. And then if we look at the most important thing for us, basically the ear, we see again 3 rows of outer hair cells, 1 row of inner hair cells. And if we look at AV1, we really just get very few inner hair cells here and here. However, if we compare that to ANC80, we do get quite a lot of inner hair cells actually. However, no outer hair cells, which kind of was the main result in the mice, right? We did not really see that in the animals, in these rhesus macaques, so in these monkeys. This is just a quantification of these inner hair cells, and as you can appreciate, ANC80 seems to outperform AV1. However, over the last few years, last couple of years actually, there were several papers that came out from other groups that used different promising AAVs, or synthetic AAVs specifically. And you can see, based on all the different green cells, that there's really a lot of cells that are being transduced. And these are all mouse experiments, and you can see that all of these 4 vectors really have remarkable potential, and not just transduce the inner hair cells, but also the outer hair cells. And 2 of these vectors have actually been used in monkeys as well, by another group in Boston, specifically David Corey. And what you can see here is that specifically here on the right side, again, pretty much all the cells of the organ of Cordy, so all the inner hair cells, all the outer hair cells, are being transduced or have been transduced with this virus. So it's really great now to have kind of like all of these different tools in the kind of armamentarium to be used then to treat all of these different diseases. And now I briefly want to kind of switch gears and talk about optical cochlear implants. We already heard about what cochlear implants are and how they function, but so what could we use this technology apart from treating or in addition to potentially treating these genetic diseases? You can actually use these AAVs as well to deliver so-called opsins. These are channels that can then be stimulated via laser light. And what we did here was that we created such a vector. There was a collaboration with Dr. Lee's lab at Harvard. And what we did here was that we injected these vectors into neonatal mice again, and then stimulated the inner ear with light. So we had this laser rod that I'll show you in just a second, created this cochleostomy, so this little hole in the inner ear in these mice while they were under anesthesia. And then we used this optical fiber to stimulate the neurons, those spiral ganglion neurons that eventually form the auditory nerve. And so first we had to make sure that we actually did get a lot of neurons. And so the delivery worked, and we do see a lot of green again. So many of these neurons are actually being transduced and had this opsin, and thus could be stimulated via light. And then if we use this laser light and stimulate the cells with the laser, and then again kind of detect the electricity at the level of the brainstem, then you see that there are these waves. So that's really remarkable, because that means that these animals actually heard the light, which is very exciting. And a group in Germany has really taken this way further, and what they are doing, or why even think, so it's the group of Tobias Moser, and they have actually developed together with a company these tools that, or these implants that can actually already be implanted in animals. And why even bother to create all of that? So here you again have normal hearing with the low frequencies and the high frequencies at the bottom. And here you have the electrical cochlear implant, so the current implant. And the kind of issue with that is that the electricity kind of spreads relatively unspecifically through the tissue. However, if you could actually use laser light or an LED to do that, the stimulation hopefully would be way more focused, and thus the resolution, the sound resolution, and thus also the sound perception in patients would be much better. Because some of the patients with cochlear implants, although as I said, it's a remarkable device and it's really great, and over 1 million people have been implanted around the world, many of these patients actually have issues with hearing regular music or enjoying regular music, for example. It's great for speech, but for music, it is difficult in many of these cases. And what you can see here is that that's again from Tobias Moser's group in Göttingen, is that this here is a sound impulse. So here you have the sound intensity, here you have the position in the auditory midbrain. And if you use a regular cochlear implant, a current cochlear implant, then you see that the stimulus looks very different from this here. And if you use laser light, then we have specific stimulation of the, or relatively specific stimulation. And this impulse looks way more like this one compared to that down here. And so the device that they use, the cochlear implant, actually has a 1 millimeter radius. So it's tiny, as you can imagine. So then, as one of the last things that I want to show to you is what about patient acceptance of all of these novel technologies? So if we are doing all this research, will it actually eventually, if we can bring it to the clinic, will it actually be accepted by patients? And so we asked 72 patients with varying degree of sensorineural hearing loss, or the parents in case the kids had the hearing loss. We tried to explain them in a kind of standardized fashion the different technologies, and had them complete a questionnaire. And we focused on viral mediated therapy, on CRISPR-Cas9, and on stem cell therapy. And we were actually surprised by how many patients, although there's still all these uncertainties, were willing to participate in such a trial, or actually were interested in these technologies. And we assessed over 20 additional variables, and we're looking at a lot of different things, like what could kind of contribute to the decision being made. And interestingly, older patients were more open towards all these technologies, and specifically also viral therapies. So what are the remaining clinical barriers, clinical translational barriers? So I created the slide a few years ago, and ever since have kind of been checking off the things that the field has solved altogether. So I've shown you some of the primate studies, and I think these answers have now sufficiently, or these questions have now sufficiently been answered. Another group in 2017 showed that just the injection per se of PBS back then, not a virus, did not result in any hearing loss. Then cell specificity with different promoters. So if we look at company data, then you can specifically say which cells you want to reach, which cells you want to transduce. Another issue with these viruses is the size limitation of these AAVs. So the genes that you can transport are relatively small, so 4.7 kilobases. But again, the field has developed technologies like dual AAVs, where you can kind of split the gene and put one half into one AAV, another half into another AAV, and then it combines in the cell. That has also been solved. to a certain extent at least. And now we really have to look at the different time windows to treat certain diseases, because in the animals that were injected at the neonatal level, in this Usher syndrome, for example, those hurt perfectly. However, if you did it 6 weeks later, once they were young adults, actually the degeneration had already taken place and you could not preserve these cells. So it's easier to prevent degeneration than to actually regenerate the cells. And yeah, in humans, sometimes that would mean potentially therapy in utero. So that would also make things obviously way more complicated. Suitable diseases, there's now 3 companies that look at one specific genetic mutation, so-called otoferlin. There's really not a lot of patients with it. So that would be an ideal disease to start with, basically. Usher syndrome, as we talked about, but also a benign tumor of the vestibular nerve, so-called vestibular schwannoma, that we've also done a lot of research on, and also published a paper on in Science Translational Medicine earlier this year, but that's not the topic of this talk. And then for other approaches, there's probably more research needed. These are the sponsors of this research that was carried out in Boston yeah, we're very thankful for. And obviously you can't do these experiments without so many different people, so many labs around the world that have done all this great work. And I'm very thankful to all the collaborators in Vienna and abroad. And with that, I thank you for your attention. And if you're interested in this topic, then I can recommend you 2 reviews, one with a more clinical focus, one with a more basic science focus. And now I'm happy to answer any questions.
Thank you, Lukas. Great talk. Are there any questions from the audience? I'm pretty sure there are.
Yeah, I have one. Yeah, fascinating talk. One question is, so it's obvious that electricity stimulates the nerves, right? That's clear. But why does it work with the laser light?
So yeah, it only works if you introduce this opsin so it's called optogenetics, this whole field. It's really remarkable. And you can use it for all types, specifically in neuroscience. It's really a big thing now. And what we've done in the ear basically is to use this virus to introduce these opsins into the spiral ganglion neurons, so the cells that eventually form the auditory nerve. And with that, you can then use the laser light. But again, from a clinical translational standpoint, obviously that's definitely not the first step, because you have to have gene therapy that works, and you have to have a device that actually is able to carry out these stimuli. But still, they were able to do it in animals. So it's really remarkable that all of this works, and really exciting.
Okay, cool. Yeah, I missed the options part. Yeah, that's clear. Yeah. That's gonna be another Nobel Prize, I think, optogenetics.
Yeah,
it's pretty clear. Yeah, it's just a question when. And did
okay, great.
Are the experiments done that I guess no, it doesn't matter. No, that question is obsolete now. I was going to ask about the wavelength of the lasers, but I think that's an opsin related parameter, right? So yeah.
Exactly. So several different opsins have been used in the ear, and obviously in neuroscience even more. And they vary in terms of the speed of the maximum stimulation, basically, and so on. And the one that we used was at the time one of the fastest, but now again there's multiple others. And yeah, it's really remarkable how fast things evolve in that field.
Great. My question would be, are you working on the implants in mice in Vienna as well? You showed several studies on the cochlear implants in humans, I guess. And then I was interested, are you doing the surgery on mice yourself in Vienna? And how tiny are the instrumentations of the implants for mice compared with humans?
Right. So so that the mouse is not an ideal model for cochlear implants because the middle ear, it's called bulla, but the kind of middle ear space in mice is tiny compared to, like also for its size, tiny compared to other species. So actually, as a rodent model, guinea pigs are more suitable because the middle ear compared to their size again is larger, and so you have more space to manipulate and to insert the cochlear implant. So that's something that I did before I moved to Boston, actually. So in Vienna, we always had a strong collaboration with an Austrian company, actually, that's producing cochlear implants. So they're based in Innsbruck, it's called Med-El, and they're actually #2 worldwide. There's only a few companies that produce these implants, and so they've also always sponsored a lot of research. And together with them, we were also able to establish this Christian Doppler lab now as well. And so we do not use mice for the cochlear implant research specifically, but we do use guinea pigs. We do use gerbils. So that's another type of mouse or rodent, basically. And now something that we've started maybe just a year ago or so is that we use pigs as well, specifically mini pigs as well, because the cochlea, the size of the cochlea is more comparable to the human cochlea, is actually relatively similar in terms of the fluid volume and so as well. And so for a lot of questions, translational questions, we hope that we can focus on the pig instead of the rhesus monkey, because rhesus monkey research is incredibly regulated. It is incredibly hard to do all of that. You need so many people who are involved. You need, like, we had a fully trained human anesthesiologist for these experiments, and I think one animal, like it took us several years to obtain the funding for these experiments, because one animal costs tens of thousands of dollars actually to get these from these highly regulated facilities. And so if we could do some, because I mean the really crucial question in terms of does it go to the brain had to be answered in a monkey, but many other questions in terms of delivery and fluid dynamics and so on, all of that can be answered hopefully in other large animal models, including the pig. And so that's why we are focusing on the pig here now recently, or we've been focusing on that recently.
Great. I had another question about the hearing nerve. If you're suffering from hearing loss after viral infection, it is usually not on both sides, but on one side, and some deafness comes from an infection. And maybe I'm wrong, please correct me on this. There's usually damage to the nerve. You didn't talk about this. Is there any way to repair or to support with your gene therapy complete lostness of the nerve as well?
Yeah, that's a tricky question overall. So to be honest, we don't really know in many cases what causes the hearing loss in sudden sensorineural hearing loss, for example. So the rapid drop of hearing for whichever reason. Sometimes you assume that it is because of a viral infection, but often you just don't know. For COVID specifically, my former boss actually looked at that and found out that COVID can lead to hearing loss, at least in the experimental setting specifically. But if we look at it population-wise, then some studies say that there is an increase, but other studies say that there hasn't been an increase in terms of sudden sensorineural hearing loss during the COVID pandemic, for example. And in terms of replacing the nerve, that again is difficult because some groups try to focus on something that's called neurotrophins. That's also from the central nervous system or neuroscience research in general, where you try to regenerate neurons. And some groups are now actually trying to kind of have like cochlear implant electrodes that release certain factors. So the easiest would probably be corticoids, because they kind of reduce the local inflammatory reaction, etc. But also neurotrophins are a big thing, because the idea is that maybe you can actually have the nerve target the implant and surround the implant even better, so there's a better connection, then the hearing is better. However, it's once the nerve is, if the nerve is really cut right now, there's no option to replace it in any way. It's just in a specific disease that's called neurofibromatosis type 2, another horrible disease, where patients often, or not often, but one of the hallmarks of this disease is actually that they develop bilateral vestibular schwannomas. So on both sides, they have this benign tumor that we were talking about before. In those cases, you use something, or not always, but some centers use something that's called an auditory brainstem implant, which kind of works similar to a cochlear implant. However, you don't have an electrode, but you have a whole array that you then place directly at the level of the brainstem, and then you stimulate the auditory areas there, because it's still tonotopically organized. So some frequencies are in some area and other frequencies in other areas. However, the more central you go, the worse the hearing resolution gets, basically. So yeah, it's
a very
it's a difficult
complex situation here. So I guess from the first instance, your diagnosis and to understand what's the reason for hear loss from the patient is not that easy as well. So it obviously needs some sequencing, gene analysis, and comprehensive auditory from your side first. Great. Are there any other questions?
Sorry, I just wanted to add, it's not like in a muscle or whatever, where you can just take a biopsy, or tumors wherever, you just take a biopsy and then you know what it is. In the inner ear, it's really encased in the hardest bone of the human body. So, and if you try to take a biopsy, then the patient is deaf. So there's really a lot on the diagnostic side as well that has to kind of be advanced, so we understand better what's happening within the inner ear and this kind of black box that we have right now.
Great, but your imaging system was very sophisticated. Dietrich.
Yeah, sorry, one question. What's the path towards human application and human trials and first in human? How do you envision kind of the next steps that are either missing that, or critical steps towards that point, or do you already have an idea of kind of first in human?
Right. So there are actually, or there have been human experiments at the moment, or like in the last few years already. So a study in the US used adenovirus in completely deaf patients. I briefly mentioned adenovirus. So there are some differences in terms of specifically cargo capacity. So the adenoviruses are a bit larger than the adeno-associated viruses. The adenoviruses are more of a transient expression, while the adeno-associated viruses are more thought of like a permanent expression. So that's why they would be ideal for kind of genetic deafness. However, what the groups in the US tried, so Heinrich Stecker was the kind of PI for that study. What he did was that, or what he envisioned was that you'd use completely deaf patients, and then you'd use these adenoviruses to introduce a so-called transcription factor that's called ATO1. And the idea, or what has been shown in animals actually, is that by introducing this ATO1, you can switch the supporting cells that are around the hair cells. We haven't really talked about that at all in this talk, but you can kind of trigger them to transdifferentiate into hair cells. And in animals, some of these animals that were treated with that actually recovered hearing, which was very remarkable. And in humans, he tried the same thing. Unfortunately, it didn't work. So that was kind of the first real gene therapy study. However, there are several companies now that are trying to recruit or have already recruited patients for their respective therapeutic kind of interventions. And in terms of genetic hearing loss, as I said, like otoferlin is a highly sought after disease, because 3 companies are pursuing that. And the main thing that's kind of holding the studies back is really to identify these patients. So really the big databases, the big sequencing databases, including like ours, we have 1 patient with that specific mutation, and all the big centers basically have 1, maximum 2 patients or so. And then you really have to find a patient with that mutation that where the cells haven't degenerated yet, right? So it's really for every specific disease, you have to find the right candidates, because if you, then do a study where nothing or not every single part is perfect, then you might end up with a failure, and you might kind of lose all the momentum that the field has right now, as it happened in gene therapy like a few decades ago, for example, with some of the unfortunate events that happened back then, that really set the field back for quite a while. And now, I think we're slowly getting to something great again, and including the FDA approval and EMA approval of these 2 drugs that I, for example, mentioned before. So that's really remarkable to see all of that.
Great, thank you.
Thank you.
Anyway, fantastic insights into the field, and supportive analysis and therapeutic approaches from your side. So I thank you for this fantastic talk today, and you all have a good summer, and we will see you next time in September or October for another Asthma virtual talk. Great.